The title of this spectacle is Somatic Cell Count. Somatic cells are the asexual body cells. So, the spectacle is related to the counting of these body cells or somatic cells which are present in milk sample. So, presence or absence, or to what extent, or to what count, or what is the load of these somatic cells in milk. It is also related to the quality of milk. Say, for example, increased number of somatic cells, it indicates that the animal may be infected, and due to that infection, due to that inflammation reactions, the white blood cells in the body tissues, body fluids, it increases. So likewise, somatic cells count, it is one of the indicators to decide the quality of milk and the health status of the milking animal. So the aim of this practical is to perform somatic cell count by the method of direct microscopic count of the given milk sample for somatic cell enumeration. Now, in your syllabus, there are two topics mentioned: direct microscopic count and somatic cell count. Direct microscopic count, it is the methodology for direct microscopic count and somatic cell count, it is remains the same. Only the targets may be different. In case of somatic cells, we concentrate on counting of eukaryotic cells, and in case of direct microscopic count, it is a broad term, direct microscopic count, because somatic cell count, it is also done by using a microscopic microscope and slide, so it also falls under direct microscopic count. Just for the sake of distinguishing between somatic cell count and counting of bacterial cells. Now, the load of bacteria present in milk sample, it can be determined by various methods. One of the methods is standard plate count, and another quickest method is direct microscopic count, that will be learning in the next practical. So the target of DMC is counting of bacterial clumps present in milk sample. So the methodologies are same. So this method makes use of direct microscopic count approach. So here the counting involves the counting of bacteria as well as the counting of body cells. So counting of bacteria will learn that in a separate practical. Here we will be concentrating on counting of these eukaryotic cells. So combine, this is the conclusions are drawn in combination, like it is not separately done. So somatic cell count and counting for bacterial clumps, so all together the results are combined, and the conclusion is drawn in combination. So both of these factors are useful to determine the past history of milk, that means whether the milk is stale, and to determine the seasonal differences in bacterial counts of milk. So it is important for raw and pasteurized milk, and this is one of the instant methods for determining presence of excess number of body cells in raw milk. So this method, that is direct microscopic count, is useful for the detection of typical type of bacteria which are responsible for causing infections like mastitis, like streptococci, which are predominant in mastitis. This method is also useful to determine the extent of contamination by soil microorganisms, acid formers, colon and putrefying types of bacteria. Now we'll study the procedure for the determination of somatic cell count by direct microscopic count method. The first step is to get a clean glass slide. Then, with the help of a marker, we have to draw an area of 1 centimeter by 1 centimeter square on that clean slide like this. So this is a slide, and with the help of marker we have to draw an area of 1 centimeter by 1 centimeter. So the area will be 1 centimeter square. So in this area, then whatever the milk sample that we have, so 0.01 ml, that means 10 microliters of this milk sample is taken, and with the help of a pipette it is added onto this marked area, and then with the help of sterile nichrome wire loop we need to spread that milk sample onto that particular area homogeneously. Now you see, milk is a rich, nutritionally rich medium. It contains fats, carbohydrates, proteins. So these fat globules are generally going to interfere with the counting of bacterial clumps or counting of somatic cells. Therefore, after the spreading, the slide is allowed to dry, means it is air dried, and then in order to remove these fat globules which may interfere with the counting, or they may be mistaken as the bacterial clumps, there the slide is placed in xylene. So xylene is a non-polar solvent and it is supposed to dissolve fat. Therefore, that slide is dipped in solution of xylene and it is kept there for 5 minutes. So this treatment will be very useful for the removal of fat, and after that treatment again the slide is allowed to dry. Then in the next step, again to clean that slide or to get rid of certain proteinaceous flocks which are present in that milk sample, the slide is dipped in 95% of alcohol. So this 95% of alcohol, it will help to remove xylene as well as it will help to clear the slide from interfering proteinaceous clumps. So it is kept dipped in alcohol, 95% alcohol for 5 minutes. Another advantage of ethanol is it will act as a fixative. That means it will bring about the dehydration of that particular sample which is spreaded onto the slide. So due to this dehydration, the bacterial cells, the clumps, somatic cells, it will remain stuck to the glass surface. So it acts as a fixative also. Then the slide is allowed to dry, and then it is ready for the next step. Now you see, the bacterial cells are semi-transparent, somatic cells are semi-transparent, very difficult to observe under microscope, and therefore it makes it mandatory to stain these cells and bacterial cells and somatic cells, and for that reason there is addition of methylene blue. So a saturated solution of methylene blue, it is used for staining purpose. So on slide methylene blue can be added, or in a methylene blue tub, this dry slide, it can be dipped for few seconds, so approximately for 5 seconds. So what will happen, that this methylene blue, it will stain bacteria, as well as it will stain the cytoplasmic inclusions, like cytoplasmic inclusions of eukaryotic cells like nucleus. So it will help us to identify the somatic cells or white blood cells. So these somatic cells, it may be of white blood cells type, or it may be cell debris, or it may be type of platelets, or it may be type of epithelial cells. Then the slide is washed under tap water, then it is air dried, and then it is ready for the examination under microscope. Now since we are observing bacterial cells or somatic cells, we need to make use of 100x immersion oil. 40x or 40x objective it will also do, but the resolution will be poor, and therefore for the sake of getting more resolution, we can make use of 50x ocular and 100x objective for the counting of these somatic cells. So under microscope, we have to observe this slide. So place that slide under microscope such that the objective will touch to the marked area of 1 centimeter by 1 centimeter. Then this slide is observed under microscope. This is the marked area of 1 centimeter by 1 centimeter, which is stained with methylene blue, and we are observing that under objective. So first place a drop of oil here, dip that objective in this, and count the adjust the microscopic field here, and count the number of somatic cells present in one field. Then you have to move the microscope ahead like this, in this direction. Now here count the number of somatic cells, blue color cells with a blue colored nuclear material, and record it. Likewise, again move ahead, count the number of cells, again move ahead, and then turn the position of the slide in this direction. Then again move ahead like this, count the number of cells, again like this. So in spiral fashion, you have to turn the slide and count the number of somatic cells present in one microscopic field. So approximately you can take counts from 10 such microscopic fields. Now what is this microscopic field? Microscopic field is circular. So when you observe under microscope, it appears circle. So in this microscopic field, we'll observe the somatic cells like this. So you can see here, in this case, we can see the somatic cells with stained nuclear material with blue color. So count it. Then after that, the next step, very important step, is calculation. This is the most important part in the practical. So here there is a formula that we are going to use, and that formula is written at the bottom. This is the formula, that is 10,000 into x divided by pi r square. Now, simply instead of remembering this formula, we'll try to understand how this formula is derived. So here, in this case, x, x is average number of organisms, or in this case, average number of somatic cells present in one microscopic field. Now in protocol, I have told you that you have to move your slides in a particular zigzag spiral fashion, and you have to count at least, examine at least 10 microscopic fields. So first microscopic field, count the number of cells present in first microscopic field. Move on to the next one, count the number of cells present in second microscopic field, third microscopic field, fourth microscopic field, so on and so forth. At least 10 microscopic fields you need to examine, and then take the average of that. So that average value means if it is x, then x1 plus, this will be x1 plus x2 plus x3 plus x4 x5 x6 x7 x10, sum of that divided by 10, it will be the average number of organisms present or somatic cells present in one microscopic field. This is how that will be your observation. Now, what is this one microscopic field? So one microscopic field, the area of that one microscopic field will be pi r square. This is a simple mathematics. So here, as the area is circular, it will be pi r square. So here r is the radius of what? Radius of that circle, and in this case that circle is microscopic field. So the radius of this microscopic field, it can be calculated or it can be determined by using a stage micrometer. So you put a slide of stage micrometer slide, it is calibrated, and try to see the lines coinciding the two borders of, or the radius, two ends of the radius of that circle or microscopic field. So this is how we can determine the radius of that microscopic field. It will be in micrometers, then you have to convert it into millimeters. So stage micrometer, it will be useful for the determination of radius of microscopic field. You need to remember that. Now what we have is, we have x value, that is the average number of cells, somatic cells present in one microscopic field, that is pi r square. Then what will be the, so x corresponds to one microscopic field, that is pi r square. Then 1 centimeter square corresponds to how much? So it will be 1 centimeter square into x divided by pi r square. This is a simple cross multiplication. X corresponds to pi r square, 1 centimeter square corresponds to how much? So it will be 1 centimeter square into x divided by pi r square. Now convert this centimeter into millimeters. So we know that 1 centimeter into 1 centimeter is equal to 10 mm into 10 mm. So 10 mm into 10 mm, it will be 100 mm square into x divided by pi r square. So here we have converted that into millimeters. We have to convert radius also in terms of millimeters. Now The next thing is that how much sample we have added for the observation. It is 10 microliters. It is 0.01 ml of the sample, and then it was added to an area of 1 centimeter square, that is 100 square. So therefore the number of cells present per ml of milk sample, it will be 100 into 100 into x divided by pi r square. Now from where this 100 has come? So 0.01 ml corresponds to 100 square into x pi r square. Then 1 ml corresponds to how much? That is simple cross multiplication. So it will be 1 ml into 100 square into x divided by pi r square divided by 0.01. So this 0.01, it will be at the denominator side. Therefore when it goes here, it will be 100. So this is per 0.01 ml and this is per ml. So you have to just convert this 0.01 ml to ml, 1 ml. So it will be 100 into 100 into x divided by pi r square. So this will be the count of cells per ml of milk sample. So this is the formula now. Final formula: count of cells per ml of sample is equal to 10,000 into x divided by pi r square. So this 10,000 into pi r square, this is called as microscopic factor. So the formula, the simplest formula is average number of cells into microscopic factor, and microscopic factor is 10,000 divided by pi r square. This is how you can determine the count. So in order to draw the conclusion now, so that will be your result, whatever is the count. For the purpose of conclusion purpose, for the conclusion purpose, you need to know what are the quality measures. So what are the baseline count? So if the count is less than 5 lakhs per ml, then the quality of milk is good. If it is more than that, till 40 lakhs, then the quality is fair. 40 lakhs and 200 lakhs, then the quality is poor, and over 200 lakhs, the quality is very poor. So whatever is the count, according to that you have to draw the conclusion that as the given milk sample shows a count of, say for example, 5 lakhs per ml, the quality of milk is good.